gluconeogenesis. It's basically genesis of new glucose. Genesis means birth, so we are making new glucose. And glycolysis, lysis means breakdown. So we are breaking down glucose. If you haven't already, check out my video on glycolysis so you can better understand the video on gluconeogenesis. Gluconeogenesis is the exact opposite of that. It's the exact pathway almost, because most of the reactions in glycolysis pathway are reversible. Let me draw the glycolysis pathway real quick. So we have glucose and then glucose 6-phosphate, glucose. Kind of messed that up there. Wait. Dihydroxyacetone, phosphate. Then they have a couple of reactions here. I'm not going to go through all of them because you can, if you want, you can check out the video on glycolysis. I will leave a link in the description. So then we have phosphoenol pyruvate here. And in the last step is also irreversible. Convert pyruvate. Now the pyruvate has to be converted back into glucose. And for this, we have to bypass these irreversible steps. And that's what we are going to do here. So first, to bypass from pyruvate to phosphoenol pyruvate, it has to go inside the mitochondria. So let's draw a mitochondria here. Let's draw a big one. Okay, it's not exactly the shape of a typical mitochondria. Just imagine if I cut it across the length and a cross section, it could look like this. Just leaving more space inside the matrix so I can draw all this. So pyruvate goes here. Pyruvate can easily cross the mitochondria. Pyruvates get converted to acetyl-CoA. And then we have the Krebs cycle here. We have citric acid cycle, citrate, and go through a series of reactions. We have malate, oxaloacetate. Oxaloacetate and oxaloacetate gets combined with acetyl-CoA to form citrate. This is the normal citric acid cycle. And you're wondering why I'm talking about this, because the substrates of citric acid cycle play a role here. The pyruvate gets converted to oxaloacetate. And this reaction is catalyzed by an enzyme called pyruvate carboxylase. And the oxaloacetate then has to go here and convert back into phosphoenol pyruvate. But the issue is oxaloacetate can't easily cross the mitochondrial membrane. So for this, it goes through something called a malate shunt. So basically it gets converted back into malate because this reaction here, it's a reversible one. So this is done by malate dehydrogenase. Malate dehydrogenase. I'm not sure if you can read that, but you get the gist of it. And then the malate can easily cross the mitochondrial membranes. You have malate here. Okay, we'll also make this dotted dashed line. Okay, malate. And the malate has to be converted again into oxaloacetate. And this is done by the same enzyme, malate dehydrogenase. So the oxaloacetate gets converted back into phosphoenol pyruvate. So we went through all of this just to bypass this reaction. Bit of a shortcut, I would say. Anyways, this reaction is catalyzed by an enzyme called phosphoenol pyruvate carboxykinase, PEPCK. So this reaction, then PEP can easily go back through this usual glycolysis pathway because all of these reactions are reversible. And here you get stuck at fructose 1,6-bisphosphate. The glycolysis reaction is catalyzed by PFK1. And the exact opposite of this, the reverse reaction, is catalyzed by a separate enzyme. And that is fructose 6-bisphosphatase. From here, this reaction is also reversible. Then you have this reaction, the first one, which is catalyzed by glucokinase in the liver and the pancreatic beta cells, or hexokinase. For this, the glucose 6-phosphate can be converted back into glucose, but that enzyme is in the endoplasmic reticulum. So let me draw an endoplasmic reticulum right here. So this has to go inside the endoplasmic reticulum, and that is catalyzed by an enzyme called glucose 6-phosphate translocase. So easy to remember these names because they describe exactly what it does. And inside the endoplasmic reticulum, glucose 6-phosphate gets converted to glucose, and that is catalyzed by an enzyme called glucose 6-phosphatase. And then the glucose can easily go out of the cell membrane and enter the bloodstream. Now that you understand the gluconeogenesis pathway, let's talk about the gluconeogenesis regulation. This is mostly done through allosteric modulators and competitive inhibition. So here we'll start with the first reaction, pyruvate carboxylase. This has a few allosteric modulators. We'll talk about them here. The first thing is ADP. ADP is a signal of having low energy. So that means it has used up all the ATP. So ADP is a breakdown product of ATP. And if you have a lot of ADP, that means you're low on energy. That means you need to stop gluconeogenesis, because gluconeogenesis is a very taxing pathway. It takes up 6 ATP molecules for gluconeogenesis, because all of these steps, glucose 6-phosphatase, fructose 1,6-bisphosphatase, pyruvate carboxykinase, all of these require ATP. ADP, if there's a lot of ADP, that means you don't have so much ATP in the cell. So having ADP, a lot of ADP, has to negatively regulate this reaction. And then you also have acetyl-CoA. If you have a lot of acetyl-CoA, that means you have a lot of pyruvate coming into the mitochondria, and citrate, it is not entering into the citric acid cycle. So that is a signal for a positive regulation in the gluconeogenesis pathway, because if you have a lot of acetyl-CoA which is not going into the citric acid cycle, that means it has to go back up to glucose. So we have acetyl-CoA positively regulating this reaction. And then we go to the second reaction, PEP carboxykinase. So PEP carboxykinase is mainly regulated by enzyme genetic expression of the enzyme, gene expression. And this is done by the insulin-glucagon ratio. So when you have a lot of glucocorticoids or glucagon or thyroid hormones which stimulate to increase the glucose level of blood, then stimulate the G protein coupled receptors, and in turn they stimulate production of cyclic AMP. And when you have increased cyclic AMP, it increases the production of PEP-CK enzyme, and that increases the rate of this reaction. But when you go to the last step, fructose 1,6-bisphosphatase, here you have a couple of negative regulators. If you remember, I'm not sure if I have space to draw this. Fructose 6-phosphate, wrong color. Fructose 6-phosphate could be converted to fructose 2,6-bisphosphate by PFK2. And this was a signal for having increased amount of glucose in blood. So basically, PFK2 is regulated by insulin glucagon ratio, and if you have a lot of insulin in the body, this phosphofructokinase enzyme gets stimulated, and you have a lot of fructose 2,6-bisphosphate. So having a lot of fructose 2,6-bisphosphate in the cell is like a signal that there is increased insulin in blood. So this signals a positive, this signals for the continuation of the glycolysis pathway by stimulating PFK1. But here, this acts as a negative regulation on fructose 1,6-bisphosphatase, which is the opposite of PFK1. So that's a negative regulation. So if it stimulates glycolysis, then it has to inhibit gluconeogenesis, because those two chemical pathways can't occur at the same time because they are counterintuitive. It's counterproductive if both of them are occurring at the same time. You're breaking down and making glucose at the same time. So in order to prevent that, you have opposites working here. And yeah, that's all for the regulation of gluconeogenesis. And that's all for this video. I hope you learned something. And if you like videos like this, make sure to subscribe to my channel and leave a like.